I'm going to give you a general overview about topics related to influenza, and I'm going to talk very fast and show you lots of slides. But if you just remember one thing, flu is very bad, and we as a community need to do better to prevent this major public health problem. So I'm just going to start off by saying that I don't know if it's possible to dim the lights just a little bit, at least in the front. But up at the top here, we see a picture of policemen in Seattle gearing up to go out on their patrols during the height of the flu epidemic in 1918. And then over here on the right, you have police gearing up to do the same thing in Mexico City at the beginning of the 2009 swine flu outbreak. So pandemics have been with us for a long time and continue to be significant problems. 100 years is what we're celebrating and commemorating with the anniversary of 1918, but about a third of that period of time is when I left the School of Medicine here and left Jack Haar's lab, and so 30 years is a really long time. And here I am in Sanger Hall in Jack Haar's laboratory, and I was able to find a picture of Dr. Sanger to commemorate this lecture. By training, I am a pathologist, and this is the best definition of a pathologist I have ever come across. So I will, anything that I say after that you can take with a grain of salt. So, you probably all have seen things come across your, the screens of your computers and cell phones in the last month or so about that this is a really bad flu year, that a number of healthy young people have died of flu, and tragically a number of young children have died of flu. And flu is just really in the news, not only here locally in Virginia and in the Washington, D.C. area, but really across the country. And it reminds us that flu is always a difficult threat that we have to think about. So, but if we cast back 100 years to 1918, I just want to tell a brief story. The U.S. was really gearing up to fight on the Western Front in World War I, starting in 1917, and 1918 was really engaged in the war. They had huge numbers of men being trained and shipped to the Western Front in Europe. And in one of these training camps outside of Boston, on September 7th, a soldier presented to sick call with a high fever. The next day, a dozen soldiers presented with similar symptoms. A week after that, 36 cases presented, and a week after that, almost 13,000 cases of soldiers presented with a very, very serious respiratory illness. At the end of the camp, about a third of the camp became ill, and 800 young, healthy 18 to 25-year-old men died. These are some pictures from Camp Devens. And in the 1950s, a letter was found in a trunk that was written by one of the camp doctors to a colleague on the September 29th. And I'll just read a few of the words. That the men start with what appears to be an ordinary attack. They very rapidly develop a very bad type of pneumonia, and then they develop blue spots that spreads, and cyanosis, bluing of their face spreads across, and when that happens, these men almost invariably die, that they struggle for air until they suffocate. It is horrible. We have been averaging about 100 deaths per day and still keeping it up. The most famous physician in the United States at the time was William Henry Welch, one of the founders of Johns Hopkins, a founding pathologist in the US, and when he came to inspect the camps, he said that this must be some kind of new infection or plague. And unfortunately, or fortunately, it wasn't a plague and it wasn't new. It was just influenza. The 1918 flu stands out as one of the most significant public health or natural history disasters in all recorded history. Here are some soldiers in a very primitive camp hospital on the Western Front. So here's some of the numbers. The historians have been revising these numbers for the last 100 years. And a conservative estimate of about 10 years ago was that 50 million people died in a few months, and many people now think that 100 million is a much more reasonable number. Accounting in much of the developing world has been very difficult and challenging, and will probably never have a real accounting, but in any case, the number was staggeringly large. In the US, a little under 700,000 people died. To put that into context, because these numbers are hard to imagine, in a city like Philadelphia, 16,000 people died, but 11,000 just in the month of October. So at the height of the flu in Philadelphia, 4,500 people died in 1 week. As the dean pointed out, the impact of the virus on military troops was devastating, so that over 40% of US troops that died to all causes in World War I died of the flu. So these are really healthy 18 to 22, 23, 24-year-old men. Something like 17 or 18,000 people died in Virginia of the flu in 1918, and my record suggests that about 1,200 people died in Richmond, which seemed to be about 1% of the population. So if we look at this map of the progress of the pandemic in the first 4 weeks from the beginning of September through the first week of October, you see that in week 1, it was spreading from port cities, both on the East Coast and the West Coast, suggesting that it was being imported into the United States, both from the Pacific as well as from the Atlantic coasts. By week 2, it was spreading into New England and the mid-Atlantic states. By week 3, it was more widespread. By week 4, it was spreading across the country, and by the first week of October, it was present and causing outbreaks in all 48 states. The ability of hospitals and medical personnel to cope with this much illness at the same time was overwhelming. Here you see that a temporary hospital was set up in Fort Riley, Kansas, in what seems to be a factory or a warehouse or something as a temporary hospital. Here you see a historical graph about the outbreak, which varied slightly in its timing in different cities and different parts of the world, but mostly the big outbreak occurred in the September to November time point in 1918, with many places having a recurrence in February, March 1919. A very poignant way to look at mortality of the 1918 flu is this photograph of death records from the state of Oregon sitting on a shelf of the National Library of Medicine on the NIH campus in Bethesda. And each page is a single death certificate that's registered, and they're bound by month. So you can see sort of a normal month in September, and then October, November, December, January with the flu, and then sort of back to normal thereafter. There are numerous photographs available, many of them on the internet, that you can find of images of people with the 1918 flu. There's one here with a student sick with the flu at Dartmouth College, and again, what looks like a gymnasium, but in military camps, and it was just a devastating outbreak all over here. At the top right is at Walter Reed Hospital, and you have patients out on the balcony, evidently no rooms in the hospital. People in 1918 did everything they did trying to prevent the flu, wore masks. They played baseball wearing masks. They went to the theater wearing masks. They served soup wearing masks, and civilians came together to help dig mass graves for the huge number of mortality that was observed. There were sites in American cities that I think the like of which has never been seen and hopefully would never be seen again, huge lorries of coffins to deal with so much mortality. The flu impacted hundreds and millions of people, obviously, but some very prominent people, including Woodrow Wilson, the President of the United States at the time, and the British Prime Minister, both nearly died of the 1918 flu. Wilson became so sick that he had to leave the Versailles Conference, and his absence is viewed by many historians as having contributed to the disharmony in Europe after World War I and the rise of World War II. The German emperor became ill, but not as severely as the other two leaders, and recovered. General Pershing, the commander of the U.S. Armed Forces, also became ill with flu, but recovered. Its impact on all aspects of society is hard to underestimate. Here, just as one example, its impact on art. The two most famous painters in Vienna, Austria at the time, Gustav Klimt and Egon Schiele, both died of pneumonia in 1918. Egon Schiele is shown here on his deathbed and was 28 years old, a typical 1918 flu victim in the prime of life. The Norwegian artist Edvard Munch nearly died of flu but recovered and spent about a year slowly recovering and painted a large number of self-portraits of him slowly recovering from the impact of the 1918 flu. The impact of the flu can be measured in 1918 by looking at life expectancy in the United States. And you can see that life expectancy in the US dropped by about a dozen years in 1918, all because of this flu outbreak. And the reason for that is that influenza viruses, and historically, before 1918 and since 1918, have had an age-specific mortality in which newborn infants are at high risk for severe illness and dying, and the elderly are at higher risk for dying. But people in between get the flu but are expected to recover with little mortality, although, of course, people of all ages do die of influenza. But in 1918, this W was changed to sort of a U, this U was changed to a W, and there was a new peak of mortality in young, healthy adults, peaking at about age 28. So people from about 20 to 40 had a very high risk of dying in 1918, and the explanations that we have for this are still incomplete, but this was one of the key features for the incredible, devastating impact of the 1918 virus. Because a lot of the people who died in that age range were the people who were of childbearing years, you can use this as a measure to look at the impact in places for which mortality statistics were not collected in 1918. Here is a study, not from my group, but from a colleague at Michigan, who's looking at 1918 mortality in India and using models to estimate the recovery of the population. And you see this huge divot in 1918. And his estimation is that at least 14 million people died in India of the 1918 flu, and other scholars suggest over 17 million people. So now that we've had a brief history tour of what happened in 1918, let's talk about the virus, the class of virus that causes this disease, and then what we have learned about 1918 and where we are in the future. So influenza, the virus of influenza that we're talking about, there's a group of viruses. There are several influenza viruses, but the one that we're most concerned about are Influenza A viruses. And these are enveloped viruses that have, they get a membrane from the cell that they're replicating in when they bud out. They have a couple of proteins on their surface, one called hemagglutinin, which comes in 18 different sort of flavors or alternative structures. But it has its one job to bind to receptors on the cells the virus wants to infect. The neuraminidase, or NA, comes in sort of 11 different flavors, and it has the opposite effect. It cleaves off those same receptors so that the newly formed viruses can be released from the cell and go on to infect other cells. The genes of the virus are encoded by RNA. It's not a retrovirus. It never has a DNA component to its life cycle. It's a negative strand RNA, so that its genes are an antisense, sort of upside down and backward coding, mirror image of the sequence. And that instead of being on one string of RNA, like, say, a measles virus, it's on 8 little pieces of RNA. And the consequences of this biology have 2 major consequences for how flu viruses behave. One is because it has its genes on multiple segments. If one host is infected with 2 different viruses at the same time, you can mix and match those genes and create a new virus with a new genotype that hasn't existed before. This is a process of reassortment and is sometimes in flu world called antigenic shift, and is a component to mechanism of when new flu strains get into humans. The second one is that since it's an RNA virus, it doesn't have the proofreading that when DNA replicates that it has a very high error rate. So when you type on your phone or you type in Word, you get the little squiggly line when you misspell a word. The RNA doesn't know that it's misspelled a word. It puts in errors at a very high rate, about 1 every 1,000 bases, so that every single influenza virus that's copied makes 1 to 10 or so mutations. And you live in this pool of basically every possible mutation happening at the same time. And the virus can take advantage of this very high error rate if it finds itself under interesting or novel selective pressures. So if a bird virus is finding itself in the pig's nose, maybe one of those mutations will help it replicate. If you give an antiviral drug, maybe one of those mutations will help the virus escape from that drug. If you have pre-existing immunity to an old strain of flu, maybe some of these mutations help mutant viruses escape from that immunity and spread. So it takes advantage of this very high error rate to do what it does. Flu in humans is a big problem every year. As we discussed, it's a big problem this year. But on average, outbreaks cause about 30,000 deaths in the United States. The highest epidemic deaths recorded by the CDC are about 56,000. And then there's hundreds of thousands of people hospitalized and millions of people who are infected. A friend of mine, David Moorefield, wrote out his flu algorithm. Is that, do you feel like you've been hit by a train? No, then you don't have the flu. Yes. Have you been hit by a train? Yes, then you've been hit by a train. No, you have the flu. So if you feel like you've been hit by a train and you have not been hit by a train, then you have the flu. This rapid ability of the virus to mutate to any kind of selective pressure is what drives the rapid change of flu viruses in circulating humans on a year-to-year basis and even a month-to-month basis during a season. And this is a family tree of flu viruses of one of the subtypes of human flu, the so-called H3 viruses, H3N2 viruses. And each one of these little black lines is, I wish the pointer would work, is a flu strain. And you can see that these flu strains form kind of distinct family groups each year, but that they're rapidly changing. And they're rapidly changing because the figure on the right shows a three-dimensional molecular structure of what this protein looks like. And the top part of it, with all the different colors, are all the places where amino acids can be changed. This is an incredibly plastic molecule. It can respond so quickly to changes. And because of this, the vaccine that's tailored for the strains that are to circulate have to be changed all the time. And the blue line shows how often the vaccine has been changed in the United States in the last 40 years or so, and so it's almost every year. So keeping up with this rapid evolution of the virus necessitates an enormous amount of expense and requiring people to be vaccinated every single year for the virus because you don't get lifelong immunity. And clearly we need to do better, and at the end we're going to talk about what we might be able to do better. Let me just talk briefly about the current flu season. Some data from the Centers for Disease Control. So as of last week, flu was at epidemic levels in most states in the US, that it's looking like a very bad year. Flu hospitalizations are much higher than the average. You can see this yellow line in the bottom. It looks like it's coming down slightly. Outpatient visits are still have not yet peaked with the recent data. So the flu season can last several more weeks, and it just has been a bad year. Recently, just the other day, they had a press conference to say that the vaccine effectiveness for this year was at about 36%, but against the H3 strain that's causing most of the disease, it was only about 20% effective. So it was not a good year for matching the vaccine to the virus that's circulating. This is looking at mortality curves in the US over different epidemic annual flu season years, and the red line at the end of 27 into 2018, these data are lagging. We don't have the most up-to-the-moment data. Look like it's going to be a very big year for mortality in the US. On the right is a very poignant graph of child pediatric influenza deaths recorded by the CDC, and so far this year there have been about 70 deaths recorded in the United States in children. If influenza were just a human virus, it would be difficult enough to control. But you add that a huge level of complexity, which is that influenza viruses are kind of accidentally human viruses, are probably mostly bird viruses. They are gastrointestinal viruses of many species of birds, but predominantly wild waterfowl like ducks, geese, shorebirds, terns, seagulls, but passerine birds, other birds, emus, lots of different species and even orders of birds can be infected with flu. Flu tends to be a respiratory virus in mammals, but a GI virus in aquatic birds, so that they shed this virus in a fecal-oral route and poop virus out into the water from their cloacas, shown in those anatomically in those birds there. And the thing about influenza is that its epidemiology, that it's epizoontology in birds is complicated enough, since birds are migrating and moving an enormous variety of viruses around. But flu viruses can cause one-off infections, but more importantly, they can stably adapt to many other host species, including wild mammals as divergent as bats and whales and camels and seals, but to animals of a major domestic and agricultural and financial importance to the world, including pigs, horses, dogs, chickens, turkeys, quail, etc. And then, of course, viruses can move around between animals and get to humans. So if you want to find flu viruses in birds, you have to capture wild birds, and then you have to ask them to bend over and cough. And you can find their flu virus, and they seem unfazed after this little operation, and you're released. But this is a friend of mine, Dick Slemons, who was the person who discovered that influenza viruses were in wild birds in the early 1970s. He's retired now from Ohio State, and I've had the opportunity to go out flu hunting with him many times. The diversity of influenza in birds, this reservoir of viruses that can adapt to other mammals and ultimately to humans, is really enormous. So if we just look at the hemagglutinin gene, there's 18 different sort of flavors or structures that you can see. You can put them on a family tree in different ways. And I thought that a week ago was Darwin Day and Darwin's birthday, and it's a picture of Darwin. And here from his notebook, he drew in the mid-19th century a diagram of a little evolutionary family tree about how things could be related to each other and diversify. And influenza is just the poster child model for evolutionary pressure and natural selection, the massive diversification that's occurred in these subtypes of hemagglutinin, so that between each subtype of hemagglutinin, there's only an average of about 30% homology between them. So they're incredibly divergent. So the old paradigm, if you would read textbooks 30 years ago, what you would find is that birds are the natural host for influenza virus, and that avian viruses adapt to pigs, and that these pig viruses adapt to humans and cause flu. There's evidence outside of Washington is limited for that, but that it turns out that humans actually give pigs flu much more than pigs give us flu. I've looked at this picture at the bottom for a long time, and I cannot decide which one's having a better time. But they're both really happy slobbering on each other. And we know that there is a lot of transfer of influenza viruses between humans and pigs. But ultimately, it's getting from that duck's orifice there to that lady's nose over there is what we have to ultimately worry about. Whether it's through a pig or horse or some other animal is not always clear. So a pandemic is a strain of flu that spreads all around the world, hence the name, as opposed to just a local epidemic. But in flu, it's given a very specific terminology, meaning that it's a new virus to humans. It's a virus that is now circulating in humans that hasn't circulated before or hasn't circulated for a very long time, to which no one or very few people have immunity. So we know about 4 pandemics in the last 100 years. In 1918, flu, which was an H1N1 subtype, which then caused epidemic flu until it was replaced in 1957 by the Asian flu, which was H2N2. Then in 1968 by the Hong Kong flu, which is H3N2. And that's the dominant virus that now, 50 years later, is still causing the major outbreak that we're seeing this winter. And then in 2009, there was a new so-called swine flu that originated back to H1N1. And we know that there are other strains of flu causing infections in humans who are in contact with birds, for example, in China, H5N1, H7N9, and we don't know what their pandemic potential is, and we don't know what happened historically. If you use mortality as one measure of how to compare pandemics, you find they're incredibly variable. So that the 1918 flu had an enormous mortality, but more recent pandemics have had much lower mortality, so that the 2009 swine flu had lower mortality than an average epidemic flu year, even though it was a pandemic virus and a new strain. And this is, so you can't say that a pandemic virus is going to be really virulent in people. It really depends on a variety of factors. You can go back in history. It becomes harder and harder the further back you go, but we think there's pretty good evidence that there have been at least about 14 pandemics in the last 500 years, averaging about every 35 or 36 years, but with incredible variable timing, so you can't predict when a pandemic will occur. It could be as high as 150 years and as low as 9 or 10 years. Here, there are just cite some examples of things that might be pandemics in the past. Over here on the left is an annual history in the year 1173 from monks of Melrose Abbey in Britain. They talk about Pope Alexander III had just canonized Thomas of Becket, the Archbishop of Canterbury, who had just been murdered a few years before. But down in the bottom in the blue here, it says that there was a flu-like outbreak of unheard of malignancy that spread across the land, and of this pestilence, many people died. And you think, well, if one abbey in 1173 wrote that, that doesn't really mean anything. But you find that monks in abbeys in France and Germany and Italy were writing the same thing. And so it's quite likely that this was a pandemic. Here, in more recent times, there's a description of the 1580 pandemic, where the German physician Dennis Sennert says that it spread in just 1 month throughout all of Europe and the Middle East, and then from there to other parts of the world, including to the Western Hemisphere. Already by 1580, flu was being brought from Europe to America, that it was given different names. And one of the names that in German it's called is the chicken disease, Hühnerweh, and because it reminded them of respiratory infections in chickens, and so you wonder if this is a reference to possibly being a bird flu outbreak in 1580. Difficult to know. Since it's President's Day today, I wanted to mention that George Washington nearly died of influenza pneumonia during his term in office in 1790, and Abigail Adams worried for the survival of the country if the first president were to have died. So let's go back to the 1918 flu and talk about the hunt for the virus. So influenza viruses were not known to exist in 1918. The concept of viruses as agents of infectious disease were still relatively untested in 1918. There were no isolates of the virus made during the pandemic, but because of the pandemic, people were extremely interested to isolate the causative agent. And so 15 years later, in both pigs and in humans in the 1930s, it was possible to isolate influenza viruses. But those were no longer the pandemic virus of 1918, and it was therefore impossible to study this virus. So in the mid-90s, I was at the Armed Forces Institute of Pathology on the Walter Reed campus, which has a collection of millions of tissue samples in one of the world's largest, or if not the world's largest, pathology archive at the time, going back to the US Civil War. And it became possible using some molecular techniques that we worked out in the laboratory to recover fragments of RNA from tiny amounts of fixed tissue that had been fixed in fixatives like formaldehyde and then embedded in candle wax. So in between my fingers there is just a little piece of lung tissue of a soldier who died of flu in 1918, from which some of these sections were taken so you can look at them under the microscope. And it became possible to begin to decode the sequence of the 1918 virus using this kind of material. Had a small team of people who worked on this project, especially my colleague Ann Reed and Tom Fanning, and over a course of a 9-year period, using this kind of material, we were able to sequence the 1918 virus. And this effort was joined by my now friend and colleague Johan Hultin, who is a retired pathologist living in California. At the time, he was living in San Francisco, and had an interesting connection with the 1918 flu in that he had been involved in an exhumation to try to find the source of the 1918 virus in 1951 in a small community in northern Alaska that was devastated by the flu outbreak, and the bodies were buried in permafrost in between these crosses on a bluff on the beach. And in 1997, it became possible for us to go back and do another exhumation, now using molecular techniques. So the outbreak in this little village at the time called Teller Mission that is right on the Seward Peninsula of Alaska. The pandemic hit in November 1918, and in a 5-day outbreak killed 85% of the adult population, leaving dozens of orphans. It had the same mortality curve, so that children got flu but generally didn't die, but adults did die. And here are orphans from that village and from other villages who had to survive very difficult conditions thereafter. Brevig mission is right on the Bering Sea, right across from the coast of Siberia. So here you can see the village, and this was in 2005, flying in the frozen Bering Sea. and with apologies to Tina Fey, this is one of the very few places in Alaska that you can see Russia from your house. This is Johan and I. This is at the bluff where the cemetery is, and permission was obtained for another exhumation, and some lung tissues were taken from a couple of the bodies, and one of them still had RNA of the virus in it, and it became possible using this material to fully sequence the genome of the virus. Now, this effort, as I said, it took us 9 years to do, tweaking molecular tools available at the 90s as much as we could tweak them. You'll find all sorts of weird things on the internet about how this was done. This one says that this was done using a supercomputer and fancy computer programs and stuff. And I just want to show you what these computer programs were. We're in Sanger Hall, but this is a Sanger sequencing DNA gel of a different Sanger, the Fred Sanger from Cambridge University, England, who worked out a method to sequence DNA and won his second Nobel Prize. But the effort, the way that we did this, is that it only often yielded 20 or so nucleotides of the virus in one effort, so weeks of effort to get 20 bases of the virus. It was an incredibly difficult and painful time, but I'll show you more supercomputers of how we notated the sequence of the virus, complex algorithms. And, but it became then possible after the whole virus had been sequenced, and then after a number of other investigators had developed methods to make infectious influenza viruses entirely from cloned genes in the laboratory, so that you can make actual infectious influenza viruses. And so those two technologies, the 1918 sequence and this ability, were married together, and after getting permission from the US government and very high containment labs, the 1918 virus itself was resurrected into an infectious form in 2005 to allow us to study it carefully in lab models. I wanted to show you a little bit, before we talk about the studies that have been done, very briefly just to mention some of the impact of this virus. Here on the left is normal lung tissue, which is mostly air. These are these little air sacs where oxygen exchange occurs. These are autopsy tissues, photographs taken from slides prepared and cut in 1918, in which the lungs are completely filled with blood and fluid. The lining of the air sacs is destroyed, and small vessel blood clots are present throughout the virus. And this is an indication of a kind of a primary viral pneumonia. But most people who died in 1918 actually died with a secondary bacterial pneumonia. No antibiotics were available at the time. And here you see massive untreated bacterial pneumonias with all the damage that caused. But you can also find evidence of repair, so that while people were still sick of the bacterial pneumonia, they might have begun to repair their lung from the viral damage, but it was the bacterial pneumonia that killed them. You can do tissue gram stains on these slides and find massive amounts of bacteria that look like streptococcus, different kinds of strep or staph, or other bacteria. And many of these were cultured at the time, so that 97% of people who died in 1918 had secondary bacterial pneumonias. We were able to look in the lab and look at where the influenza virus is in the tissues of people who died, and the virus replicates in the entire respiratory tract, from the nose all the way down to the air sacs lining the lung. And so the virus was really widely distributed in 1918 in these autopsy sections. And so now if we look at what we've learned from the 1918 virus, the first thing I want to say is that we've learned that all human influenza cases since 1918 are all derived from this one single new virus that got into people. So maybe 100 million people died in 1918, but that many people or more have died of influenza every year, of annual flu, and when the pandemics came in 57, 68, and 2009, all of these are just descendant viruses of this one founder virus. And so you can really think that a single introduction of a virus to humans over a 100-year period has caused an enormous amount of morbidity and mortality. And it's really amazing to think that what we are in, my colleague David Moorefield and I call maybe the possibility of a pandemic era. We're still in the 1918 era, since the viruses that caused this year's flu are still the direct descendants of the 1918 flu. So very briefly, I'm going to talk about some more basic science, so feel free to browse your phone if this is boring. But there are three factors that we think are important for how influenza viruses cause disease. There are viral factors, there's how the host responds to the infection, and then bacterial pneumonias as a secondary complication. So let's talk briefly about viral factors. So working in high containment laboratories, it's possible to work safely with the reconstructed 1918 virus and other similarly hot viruses. And unlike human viruses, if you took this year's flu strain and you gave it to a mouse, very little would happen. It might lose a couple of % of weight and then recover. But with a very tiny amount of virus, the 1918 virus, without any further adaptation, kills mice, kills ferrets, kills multiple species of monkeys, is a very, very virulent virus. And the black line shows weight loss in mice. At 4 days, mice have lost 30% of their weight after being infected with the 1918 virus. We have done a lot of work to try to understand what the virulence factors are in the virus, and the major virulence factor is the hemagglutinin gene, the surface protein gene. So if you put that onto a modern seasonal flu that has no mortality in a mouse, you can get a virus that kills mice. So just that one gene out of the 8 is accounting for most of the mortality of the virus. The amazing thing is that what we had hoped is that we would find mutations that were unique to 1918 that would help us understand that mortality. But the scarier reality from that is that it looks like that this virulence is something that it just inherited from a wild bird ancestor of that subtype, of an H1 subtype. So that is, if you can take a wild bird virus, H1 hemagglutinin, and put it into the 1918 virus, it doesn't attenuate it at all. If you put it on the background of a modern human virus, it kills mice and ferrets in a way just like 1918. And it suggests that virulence was not selected for, and this kind of makes sense. The virus replicates so quickly, it's kind of a blitzkrieg virus. And it's hard, obviously the virus is not thinking, it doesn't have intention, and I'm going to apologize for thinking anthropomorphically. But I don't think the virus, in a sense, cares about subsequent damage to the lung. If it replicates in a couple days and it's coughed and sneezed out to someone else, and you subsequently 8 days later get a bacterial pneumonia and die, there's probably very little negative selection for that virus. What is important for the virus is, can it replicate in a human cell? Can it be spread from a person to a person? So I think there's huge selection for that, but I think virulence is probably not selected for. And the scary feature is that bird viruses have features like 1918, and we wanted to know which ones. So if you take all the different hemagglutinin subtypes and make a series of viruses in which the only gene you change is that coding for the surface protein, and you evaluate that in mice, you find that most of them cause no disease in mice and ferrets and other animals, and a small handful of them cause a lot of disease like 1918, subtypes H1, 6, 7, 10, and 15. And scarily, the way the host inflammatory response behaves is very much like 1918, and it doesn't seem to be randomly distributed in the flu family tree. The ones with showing the lethal infection in mice are in related groups of flu viruses on the family tree, so they probably share some structural features. This is not just an artifact of lab animals. If you take these same viruses and you culture normal human tracheal epithelial cells, you can get them to grow in culture so that they make cilia on the top and produce mucus. And if you put these viruses into these cultures, you find that all the viruses replicate to about the same level, but that the H1 and H7 viruses, for example, rapidly kill off the cells of the culture, where the H3 and H viruses do not. And here you can stain for where the virus is. The green is the virus getting into all the cells, whether they're ciliated cells or mucus cells, but just not into the basal cells, interestingly. The scary thing for me is that there have been outbreaks of viruses in China particularly, in which bird viruses of those same subtypes have been causing disease in humans, and one of them that worries me a lot is H7 and 9, and I'll talk about that in a minute. There have, in the last 4 years, there have been outbreaks with H7 and 9 and at least 1,600 confirmed cases with at least 620 deaths. So it's about 38% mortality. This virus has not been able to adapt to humans and cannot spread from person to person efficiently, thank goodness, but has obviously very pathogenic properties. Here you see it spreading in waves, and recently we've been able to sequence viruses from isolates from China, and there is an enormous amount of mutational variation going on in hemagglutinin and other genes. And here you can see on the top a model of hemagglutinin where some of these changes are. The US government commissioned that a vaccine be made and put in the national stockpile after the 2013 outbreak against the strain in 2013. But because the virus has evolved so much in these last 4 years, the viruses currently causing disease in China are no longer a good match for this 4-year-old vaccine. So the tens of millions of dollars that were spent on stockpiling the vaccine in 2013, probably not very useful for us, and a new vaccine will have to be made to try to keep up with this rapid evolution of the virus. Just something else in the news last week is that the swans next to Windsor Castle, the Queen's swans, most of them were killed off in an outbreak of a high-path bird flu, an H5 outbreak. So this is something that happens all over the world and is important. Briefly, let me talk about host factors. When you're infected with a virus, your inflammatory response and the subsequent immune response is a contributor to what happens in terms of the damage and an immunopathology, that is, how your own immune system could potentially damage your lung. And in the 1918 virus, something really unusual and remarkable and awful happens, which is that after infection, you get a huge infiltrate of white blood cells into your lung, but the most common cell that comes in are neutrophils. And you get a huge upregulation of genes associated with an acute inflammatory response. This is a gene array. The ones in bright red are turned on, and the ones in green are turned off. And the 1918 virus, compared to other viruses, turns on this inflammatory response and turns on cell death receptors. And using this information, we looked at a drug that has no effect on the virus, and we gave mice a lethal infection of the 1918 virus. But half of them were treated with a drug that blocks one of the byproducts of these white blood cells so that it doesn't release free radical oxygens that cause a lot of damage. And mice that didn't get the drug all died with bad pathology, and mice that got the drug survived even though they had a lethal dose of the virus. But the viral titers in their lungs were not lower than the control. So the virus itself isn't the only thing that's causing damage to the lung. The inflammatory response is causing a big factor. And drugs like this might be able to be used in severe infections like H7N9 to help mitigate disease. Lastly, let me mention the bacterial factors. I mentioned that almost everyone who died of flu in 1918 had a secondary bacterial pneumonia. But even today, a very large percentage of people who get viral pneumonias can develop secondary bacterial pneumonias, and controlling them in an era of antibiotic resistance becomes very challenging. But one of the things that we've learned about what flu does in terms of secondary bacterial infection is that flu viruses tend to infect the cells just on the tips of the cells that line your respiratory tract. But in the case of the secondary bacterial infection, the underlying cells are exposed to bacterial attack and are killed off. And so the stem cells of your lung, the cells that can actually regenerate and help you rebuild your lung after an initial damage, are lost. And so what you have is an inability to reproliferate the epithelium that lines your respiratory tree. These basal cells are lost. And so what you have is really an inability to repair is one of the key mechanisms of damage in secondary bacterial pneumonias. And in 1918, the situation is even much more complex in that the bacteria that infect the lung after the viral infection respond to the white blood cells there and the inflammatory response in a way that makes the bacteria more virulent, and that you create this vicious cycle in which both the bacteria and the virus are stimulating white blood cells and causing a lot of damage. And one of the things that happens in addition to damaging epithelial cells is they damage blood vessels, and you get a lot of blood clots forming. And the 1918 autopsies are full of blood clots in the lungs, where you're not seeing that very commonly in autopsies of people who die of flu viruses in other years. And here you see one of those blood clots down in the bottom right. There's just a very quick aside story is that one of these pneumonia cases in 1918 with the blood clots had blood clots that looked very interesting with very sickle-looking red blood cells. And this case turned out to be an African-American soldier who got sick with flu and died with a bacterial pneumonia in the fall of 1918, but who had the sickle cell mutation, the classic mutation in hemoglobin that led this person to have sickle cell disease. So this actually turns out to be the oldest diagnosed case of sickle cell anemia 4 years before the term was even coined in a paper in 1922. and obviously long before the mutation was understood. But clearly the impact of flu on someone who had an ability to have this kind of clotting was obviously must have been devastating. This effort to sequence the virus in the 90s, this 10-year effort to sequence the virus is just too painful to imagine doing again. And we're trying to find viruses from before 1918, and I'm not young anymore, so we need to do it faster. So technology has caught up with us, and recently in my lab, we've developed some methods using some next-generation sequencing technologies, where in one sequencing run, in about a 2-week effort, we sequence the complete genome of the 1918 virus from a 1918 autopsy, plus really good expression of host inflammatory genes, plus the transcriptional factors of the bacterial co-infection. So we got the whole kind of environment of a fatal 1918 infection in one sequencing run. So luckily for us, as we move forward to try to find older cases from before 1918, it's not going to take as long. So in the last couple of minutes, I just want to turn toward where we're going in the future, and that as we think about pandemics, we have to think about seasonal flu and vice versa. And one of the things that we're doing in my group at NIH is to actively study influenza in people. So studying influenza in lab animals is certainly interesting and informative, but ultimately as physicians, we're interested in how influenza causes disease in people. And a cornerstone of this effort is our influenza challenge model, in which carefully screened healthy volunteers are brought into containment wards in the hospital and given currently circulating wild-type influenza viruses, and their self-limited influenza is studied, and their inflammatory and immune responses. And then this is used as a basis for phase 2 studies to look at novel vaccines or drugs. If you look at these curves, the black line is how much virus is shed out of the nose of someone infected with flu by day, and the dotted line is the symptoms they develop. And what you can see is that for the first day or so, you can be shedding lots and lots of virus, but have very little to no symptoms. And this is how flu spreads so well, that you don't know you're sick. You get on the subway, or you are talking to friends in the office, and you might be shedding virus, but you don't know you're ill yet, or have only very mild symptoms. You don't really recognize you have the flu. But this gives us an opportunity to use this very short time window of a day or two to see if we can develop prognostic or diagnostic markers looking at gene expression of white blood cells in the blood in this window. And interestingly, by day 1 or day 2, it's possible to stratify patients as to how long they're going to shed virus. So the ones with the genes on the red genes on the right are, by day 2, we can make a very strong prediction that they're going to shed a lot more virus than the people who do not have that gene expression pattern. And that here's another stratification of people who are going to have the most severe disease over the next week by day 1 or day 2 can already be predicted. But more importantly, for future vaccine development, we can make very good predictions now by about day 4 or day 6 as to who is going to develop a really good antibody response against their virus. So if we want to evaluate a new vaccine, we can say at day 4 if they're going to have a big rise in antibodies against this virus at 2 or 3 months after their infection, which is very exciting. So in the last minute, I just want to say that the kind of influenza vaccines that have been made for the last 70 years clearly work when the virus that circulates matches the virus that's put in the vaccine. But because it takes at least a year to make this prediction, to manufacture the virus, and flu changes all the time, as we're seeing with this year, this is not a good long-term strategy. And so the sort of dream of the flu community and of the public health community would be a vaccine that had broader protection. So they could be small steps. If you had a flu vaccine that would provide coverage for 5 or 10 years and you wouldn't have to be vaccinated every year, that would be an enormous help. But if you could have a vaccine that would prevent for future pandemics, if it was a bird flu or a horse flu or swine flu, whatever it was, if it could be prevented, that would be remarkable. But it's hard to know if that could actually work in people. A number of people have ideas about how to move forward with this, and some very exciting ideas are being tested now in the lab and will be soon tested in the clinic. And in our group, we're looking at some ideas to how to make a universal vaccine based on structures initially determined from the 1918 virus, but looking at a cocktail of vaccines that express hemagglutinins selected from a variety of bird flu viruses. And just to show you in some animals like mice or in ferrets, here you're looking at mice that have been either vaccinated with this vaccine or have been given salt water, and then they come back and they're challenged with a lethal dose of a virus. And so this is weight loss. And so the animals that only got salt water, no vaccine, they rapidly lose weight, the dotted line, and 100% of the mice are dead at day 7, whereas the vaccinated mice lose no weight and are fine. But this is a matched virus. So this is a virus that matches what's in the vaccine, so that is not really surprising. What's really remarkable that we've seen in mice and now seen in other animal models is that viruses that are not in the vaccine at all, completely different subtypes. So, for example, the 1957 H2 or an H11 bird virus, when given at lethal doses, also show 100% survival in our models. So in our initial version of the vaccine, we had 94% efficacy following 10 times lethal dose challenge with 8 different subtypes of flu. In our newer version of the vaccine, we're now seeing 100% efficacy with really good results in ferrets. And so we are getting into phase 1 clinical trials in humans within about a year to look at safety and the immunogenicity of this vaccine. And we're very excited that this is a non-infectious delivery, probably intranasal, and with no adjuvant, and might provide very broad protection. So it would be interesting to see what will happen with this as we develop it. So in summary, flu is a huge public health problem. Its pathogenicity is really complex. And cannot just be ascribed to just what's happening in the virus or what's happening in the host, but a combination of those things. That the 1918 virus shares its major virulence factor with currently circulating bird flu viruses, and that studying flu both in animals and in the lab is important, but studying them in humans is crucial to help us develop new vaccines. So I will end with this, and then my acknowledgement slide says people in my group, and then just say funding from the intramural program of NIH, funding from HHS Central, from BARDA, from DARPA, and from the Bill and Melinda Gates Foundation. So thank you very much for your attention.
That was just spectacular. Absolutely spectacular. Thank you.
That was just a spectacular presentation. Thank you, Jeff. We have a brief panel that will be led by our VP for Research, Dr. Frank Trina, and will include Dr. Michael Donnenberg, Senior Associate Dean for Research, and Dr. Dave Wenzel, Professor Emeritus. Would you like to come forward?
Okay.
Okay with you, Jeff, we'd like our panelists, Dr. Wenzel and Dr. Donnenberg, to start out with a couple of questions and then open it up for a general discussion and questions from the audience. Dick?
Well, first of all, in this Olympic season, that was Gold Star. Thank you very much. So my question is this. If you're in the business of recreating the genome of an entire virus, but one that actually is incredibly deadly, many of us might ask the question, well, what if? And so my question, well, I'm sure you've thought about this, how do you deal with the ethical question of the what if? Mm-hmm. What questions you ask yourself, and how do you approach the response to the ethical question that's obviously out there?
Yeah, no, it's an excellent question. And this is something that was thought about a lot from the inception of the project, and all the work was done thinking about this and its the containment and weighing the positives and the negatives. So you have the positive of what science could be learned to help trying to prevent something like 1918 from ever happening again. You have the risk that either an accident could happen and virus could be released into the community, or someone intentionally could use information in a nefarious way. And this is a problem, obviously, or something to be considered for what we're doing, but is really something in almost all of technology. That is, almost any technology can be developed for positive and then have a negative connotation. The US government spent a great deal of time considering these questions. The decisions to reconstruct the 1918 virus literally were cabinet level decisions, and we had proceeding with the approval of the Secretary of Defense and the Secretary of Health and Human Services. The work was done in high containment labs under extremely careful conditions at the CDC initially, and then when the labs were built at NIH. To work with these viruses, you have to register into the Select Agent Program. Everyone who works with them has to have very stringent background investigations, FBI investigations, continuing ongoing medical and psychological evaluations, lots of biosecurity, physical security. To get into the lab, you have to have retinal scans, all sorts of things to make sure that the engineering and the technology is such that no inadvertent escapes would occur. Of course, the sequence of the virus is published in GenBank, and the ability to actually make and synthesize small genes and put them in plasmids and actually rescue viruses is not difficult anymore. It is possible. So I think that everyone in the field has struggled with this, and that the US government put a moratorium on what they called gain-of-function research in 2012, and that is that you would put a moratorium on working on anything that would enhance the virulence of an agent or that could enhance its transmissibility in a mammal. But since flu viruses in nature are doing that continually in an unregulated way, ultimately, after lots of thinking, it was decided that doing this work carefully and in controlled ways in which the experiments are proposed, that they are evaluated, and those that are deemed to be reasonable are done under this kind of scrutiny. So can I say that there's no risk to this kind of work? Absolutely not. But I can say there's absolutely a huge risk to us if we don't help mitigate future pandemics.
Thanks very much.
So ever since 2003, or in the years following 2003 when H5N1 first appeared and it's so deadly, I heard many experts say it's not a matter of if this becomes pandemic, it's a matter of when. And as everybody said that right up until 2009, when we were hit with an H1N1 pandemic. Why is it that we're so bad at predicting what the next pandemic will be? And what can we do to improve that?
Yeah, no, that's also a good question. It's very humbling that a virus with 13,000 bases and 8 genes is a heck of a lot smarter than lots of really dedicated human beings. The virus is just so unpredictable by its very nature, its massive, rapid evolution, that I think the only thing we can ever do is try to chase behind it. I think it's very hard to make forward-looking predictions. People have tried for 40 years now just to predict the kind of drift that would occur in seasonal human flu from one year to the next. Could we predict in advance using computational methods what the mutations are going to be so that we could make the vaccine in advance? And those have all just been inadequate. The virus is so amazingly plastic, it's just hard. Obviously, we need more surveillance. We need more surveillance at the human-animal interface, that in places where they have live animal markets in big cities like China, where you have the risk of zoonotic infections like flu or coronaviruses like SARS, that doing things to mitigate how they're cleaned out and how they're regulated is also important. But at the end of the day, I think that we're always going to be at the mercy of influenza viruses in nature to be able to jump species and adapt and do things that we can't predict. It's our effort to see if we can do something that would help make a kind of a new class of vaccine or several different kinds of vaccines that might help at least mitigate that. Maybe not prevent it, but certainly mitigate the impact of a new pandemic. Luckily for us, H5 hasn't adapted to humans, and I'm not sure that it ever will. I think that if you look at viruses that are very adapted to chickens, this is really just my opinion, but I think there's a number of data points that support this, is that viruses that get adapted to chickens are kind of a dead end in terms of mammalian infections, and that we're really at more risk from avian viruses in waterfowl like ducks. There are certain mutations that occur that are characteristic of viruses adapt to chickens that we never see in past pandemic strains. So I am thinking that H5 might be really a supremely adapted chicken virus, but not have that much risk to adapt to humans. where H7 is a virus that's in multiple species, not just chickens, but is in other wild birds, as divergent as pigeons and songbirds, and it's not clear exactly where it is, but it's also scarier because the H5 virus kills birds, and so you can find where it is because flocks of chickens, 100% of them drop dead, where the H7 virus is a silent infection in birds, and we don't know where it is because the animals are not ill. So it's an even more deadly risk, I think.
Audience questions?
The microphones.
They're coming with microphones.
Clive Baumgarten. I was thinking about your side issue, the use of an SOD mimetic. And of course, you were using it to reduce the amount of superoxide that was available, which is very damaging. But the other side of that chemistry is that it increases hydrogen peroxide. Is there any evidence that that might be a component of its beneficial effect? And is there any thoughts about including, something like a manganese SOD mimetic in as part of a procedure for treating patients?
Yeah, that's a good question. So the drug that was used is a saline manganese compound. It's a catalase and superoxide dismutase mimetic. So, I think that in an unusual circumstance like this, in a viral infection inducing a big neutrophil infiltrate in which you got a big free radical oxygen response, could be useful. But since one of the major consequences of a severe flu infection is to be set up for a bacterial infection, what would be the consequences of using such a drug in the context of a bacterial pneumonia, right? So these are, this was mostly done as a proof of principle to show that the host inflammatory response itself is a component to the disease, but there is the possibility that in a very tailored circumstances, such a drug might be useful, say an H7 infection in which there isn't a secondary bacterial pneumonia, but it's just way too premature to speculate on its really therapeutic use.
I was wondering, just following up on that, some of the Harvard guys have been playing around with modulating the neutrophil infiltration with arrestins and defensins. And also here at the medical school, Dr. Fowler has been looking at high-dose vitamin C for sepsis, but he's also thinking about it for ARDS. Have you thought about modulating the infiltrate as well as dealing with the potential value of vitamin C?
No, those are very interesting points. We've certainly thought about those things. The last few years, the direction of our lab is focused more on the vaccine and not on those studies. We would like to get back to this kind of work, but I think that thinking about immunomodulatory treatments for infectious disease in general is a really amazing topic with a lot of future potential.
Thank you for a wonderful talk. Do we know what's responsible for the flu being seasonal? Why does it occur just during winter months?
Yeah, that's an excellent question. So in general, flu is seasonal so that the northern hemisphere and the southern hemisphere, which have 6 months opposite winters, tend to have opposite flu seasons. So during our summer is Australia's winter flu season. In the tropics and around the equator, interestingly, flu is much less seasonal and can be found at many times of the year. It doesn't have a distinct seasonality. Many people have speculated about this. There are a number of factors. There certainly questions of temperature and humidity probably play a role. You have an enveloped, very fragile virus that's being spread in small and large droplet aerosols. So on high heat and low humidity, they desiccate and don't remain infectious. So that's a component. I think, frankly, another big component is that after September, kids go back to school, and kids are an incredible vector for flu viruses. I said that birds are the natural host of flu. Anyone who's a parent can tell you children are the natural host of all viruses. And if you look at 2009 as a great example, there was a small outbreak that occurred in the spring. Pandemics, when strains go from animals and get into humans, there's no seasonality. It just happens when it happens. And then within 1 year, though, they settle into this winter seasonality. After 1919, it became seasonal flu. But the 2009 pandemic caused a little outbreak in March and April in Mexico City in the southwest US, and there were outbreaks in New York City, small outbreaks. And then the CDC started surveying for the virus, and then it basically went away, and it was under the radar, And right after Labor Day, the pandemic peak went zhoop, just like that in the first week of September. And it wasn't cold in most places in the US. I'm thinking that kids played a huge role in that.
I'm not a doctor, I'm an actor. I'm not a real doctor. Two questions. One, is there any evidence of people that survived the flu passing on a gene that led to Parkinson's disease? And two, where did it start? I've read that it started near Fort Riley, Kansas, and spread. So those are my two questions.
So let me answer the second question first, and that where did it start? I am officially and completely agnostic about where it started. There are lots of people who have their favorite theories. The classic kind of American history view is that it started in Fort Riley, Kansas. But if you look at the map I showed you early on of where flu was being reported in September, it was in San Francisco the same week it was in Boston. And it was not in Kansas. And the outbreak spread from the ports along the rail lines, exactly along the rail lines. So I think that by the spring of 1918, the virus was already globally distributed. I think that it started circulating in humans in the season of flu before, that is the winter of 1917 to 1918. But the earliest case we found is of a case of the 1918 flu of a soldier who died on May 11th, somewhere in the Midwest, I don't remember where. But we haven't been able to go further back in time. But I think that the virus was percolating along under the radar. And if you think about a virus that spreads with an infection rate of about 1.6 or so, that is, one person with flu can spread the virus to about 1.6 people. Maybe 1918 it was a little higher, maybe it was 1.8. But it's nothing like measles, where you walk into a room with measles and you can spread the virus to 20 people. So it spreads slowly, and at the beginning, those numbers are just so small, they're just under the radar, and it takes a while for the kind of exponential increase to occur. So I think it didn't become apparent, but I'm sure it was circulating, and there's evidence that it was in Scandinavia in the spring. There's evidence that seems to suggest that it was in northern China in March of 1918. So I think its origin is backward in time, and we'll probably never know the answer. The second question about the relation to Parkinson's is a very interesting one, that there was an Austrian physician, von Economo, who described an outbreak of an acute infection of the brain, an acute encephalitis, and there was a reasonable mortality, but of those people who recovered, they developed very early onset, very profound Parkinson's disease. And the film The Awakening is one of these patients, one of these post-encephalitic Parkinson's patients. That outbreak occurred starting in about 1917 and during the flu and spread. It peaked in the early 20s, and somehow I think they've been kind of conflated. We looked at autopsy tissues of people who died of encephalitis lethargica in the 20s, about 9 or 10 cases. It's pretty hard to find autopsies of people who died. And they were all negative for influenza, and you can't prove a negative, but I don't think it fits epidemiologically. I don't think it's the flu. I think it was its own thing, but it was associated with profound Parkinson's.
We have one more question, just one second. I'm coming, wait.
Thanks. So if the virus mutates so fast that we can't predict what it's going to do, you care to speculate on the mechanism for the possibility that it's actually able to reduce mortality in the ferrets and the mice that you kind of briefly hinted at those last couple slides?
Yeah, so we're working out the correlates of immunity in these animals, and what we're seeing is that it's both B cell and T cell immunity, that the way that we're presenting hemagglutinin is generating a lot of a T cell response, CD8 specific T cells, which are unusual. That it's not that flu infection generates a T cell response too, but generally not to the hemagglutinin protein. It also generates antibody responses, including to conserved areas, to the stalk or stem of the hemagglutinin. So it's clearly a blended response. Our goal is not to develop what I think you would call, say, a neutralizing or sterilizing vaccine, so that you would not be infected at all, but that what you don't have is severe disease. So if you can drop viral reduction 3-4 logs in your respiratory tract, one, you're probably not going to shed so much virus. Two, you're not going to get pneumonia, you're not going to be hospitalized, hopefully. And three, every time you get the flu, in a sense, you're being boosted, in a sense, maybe. But the real kicker is that when you look at naive animals who've never seen flu and are being given these vaccines and then challenged, do not recapitulate what the human population is, which is people with decades of exposure to all sorts of different viruses. And so we don't know actually if we're going to see responses in humans with a kind of a complex history of exposure to flu that we do in naive animals. And the only way to look at it in humans, we're trying to replicate that in animals like ferrets that live a long enough time that you can infect them with H1 viruses and let them recover, then infect them with H3, and we're kind of creating a repertoire. But at the moment, we don't know the answers to those questions.
I think we're going to draw to a close. I'd like to thank our panel for their questions. I'd also like to point out that in our audience is a Miss Haar. And if you remember back to the beginning of Jeff's presentation, he said 30 years ago here, he trained in the lab of Dr. Haar. There is Dr. Haar. Fantastic. I'm glad I didn't pronounce you dead or otherwise. So congratulations to both of you. As a medical school, we train doctors to go on and do great things. And I think Jeff has given us an example of what an incredible training can have impact on health and wellness. I'd like to thank you for an outstanding presentation. This is a small gift of appreciation. It's actually a BMW car. You just add water and it worked just perfectly fine, Jeff. Thank you for a great presentation. Thank you.